Part 2 of Chapter 10's discussion of molecular biology of the gene. With the basic knowledge of the steps of gene expression behind us, we can now go into transcription and translation in a little more detail. So we'll move over to section 10.9 for transcription. Now this little drawing up here shows a portion of the DNA, and of course we're going to make a copy of one of the genes of DNA. Now there's an enzyme called RNA polymerase which is involved in transcription. It doesn't just start anywhere, but it locates a portion of the DNA called a promoter. That's basically the area that says, okay, it needs to start here. So as RNA polymerase slides down the molecule, it unzips the molecule and rezips it. But while it's unzipping, it exposes the DNA bases so that RNA bases can be assembled according to the base pairing rule. So as this goes down, it's making a copy of the gene, and then when RNA polymerase reaches the termination site, it's over, and we now have our single-stranded messenger RNA molecule, which is a copy of the reading frame of DNA. Now, this will leave the nucleus, and it will go into the cytoplasm and then go to the ribosome. Now, in the previous video, I forgot to mention that translation occurs at the ribosomes. That is the site of protein synthesis because it is the site of translation. Now, we're at the ribosome now for translation. Now remember, we already covered how the genetic code translates, but we didn't talk about the interpreter. The interpreter is this molecule here called transfer RNA, or tRNA. It's a single-stranded molecule which folds over on itself sort of like a clover leaf. On one side of the molecule, you have a 3-letter anticodon which recognizes the codon, again, by base pairing. And on the other side, it carries an amino acid piggyback. So the first transfer RNA will come in, bring in the first amino acid. Then the second transfer RNA will come in, bring the second amino acid. Then the third transfer RNA will come in and bring the third amino acid. Meanwhile, we're growing our protein one amino acid at a time. Then when we reach the stop codon, it's over. We have our string of amino acids which gets configured into a protein. This happens at the ribosomes. Now, section 16, you need to look at the pictures in your book. It discusses mutations. Now, it discusses a substitution, an insertion, and a deletion. A substitution is changing a letter for another letter. Now, if it's within the redundancy of the code, then it will have absolutely no effect because it will code for the same amino acid. However, if it changes the amino acid, then our amino acid sequence is different by one, and it could have an effect on the protein. One example is sickle cell hemoglobin. It only differs by one amino acid caused by one base pair substitution. Now the other two are different. Insertion means you're adding a new letter, and a deletion means you're taking away a letter. So either way, what you do is you shift all of the letters over by one, and all of the codons from that point on will be different. So those are types of mutations. Now, we skip all the way over to section 20 and has a brief discussion of HIV. HIV is the virus which causes AIDS. It attacks helper T cells. Now HIV is known as a retrovirus, which means that instead of DNA, it has RNA. Now what a virus has to do is it has to squirt its genetic material into the cell that it invades, and that genetic material needs to splice itself into the cell's own DNA, and the cell does all the dirty work for it. Now, in the case of HIV, it's an RNA virus, and RNA has to be spliced into DNA, but it can't be done directly. It has to be copied onto DNA, which is the opposite of what happens ordinarily. So what you have is you got RNA going to DNA. That's called reverse transcription. Now, there are enzymes called reverse transcriptases that catalyze this, and some of the medications that are used to treat HIV/AIDS are reverse transcriptase inhibitors, blocking this backwards molecular machinery and hopefully stopping the virus from replicating. And that concludes the part 2.